It's my pleasure to introduce the next speaker. The next speaker is Dr. Helena Gellersen. She's going to be speaking about the distinct contributions of dissociable brain networks and hippocampal subregions to memory fidelity.
Thank you so much. I'm very pleased to be here. I have no disclosures. And so some of you might have a very strong feeling of déjà vu right now. And that is because I also gave a talk at the symposium this morning. And this actually very neatly brings me to the topic of my talk, namely that of pattern separation. Now, most of the regions in your brain are likely to have highly similar neural responses to a lot of the features that are shared between these two talks. And so ideally what would happen in your brains now after this talk or during the talk is a process deemed pattern separation, where these neural responses are going to be decorrelated for you to later on be able to differentiate them. And this is crucial for high fidelity memories. And we've already heard a lot about entorhinal hippocampal circuits today. So we know that different hippocampal subfields are arranged in this particular trisynaptic pathway, and the idea here is that the dentate gyrus might be specifically acting as a bottleneck for pattern separation. So input coming into the dentate gyrus being decorrelated and then being passed along to other regions. So being termed a universal pattern separator. But it's actually really difficult to tell this apart from CA3 signals in the 7 Tesla scanner, in the 3 Tesla scanner. So ideally we would want ultra high resolution imaging because it's not been shown as clearly that this universal pattern separator profile is actually the case in humans. Another question is that, of course, the hippocampus receives a lot of different cortical inputs. And these inputs are coming from different brain networks that have been identified as having different selectivity profiles towards object and scene information. So what we did is we used a mnemonic discrimination task, which is often used to get at pattern separation. So the idea here is that you're always being presented with new targets, and then after that, you're either being presented with an exact repetition of that target or a lure that has some shared features to varying degrees of feature overlap. And we did this in a 7 Tesla scanner with healthy younger adults and a 1 millimeter isotropic resolution for the functional image. But what counts as evidence for pattern separation? So a region that already decreases its activity very strongly when a low similarity lure is shown, that is considered not particularly well placed to support pattern separation because it is very sensitive even to low levels of feature overlap. But a region that maintains its activity pattern throughout different lower similarity bins is actually capable of this type of pattern separation. So this is really what we're looking for here. But what is a high similarity lure? What factors drive this? So is it just the behavioral readout, so memory difficulty, for example, or are there perceptual and semantic features that might be sufficient to explain this even without getting a behavioral readout? And this is one of the questions that I wanted to answer here. Specifically, first, which regions are involved in pattern separation and other contributions domain specific? How do different stimulus features modulate the response of various brain regions during pattern separation? And a very quick look at our preliminary results will be whether we can use multivoxel pattern analysis to reveal highly dissimilar patterns of lure and target representations in the dentate gyrus. So how do we define stimulus similarity? We use 3 different types of neural networks to do so. And the first one is the one that has been previously mentioned, so the CLIP method, which was developed by OpenAI in this case. And this one uses pair text and image encoders. And this means it's incorporating semantic abstraction and language-mediated similarity. We also used DreamSim, which is trained on perceptual similarity and confusability ratings that were obtained from human participants. So this means that this assesses image similarity on a more holistic method, and thus potentially more closely might align with human psychological aspects of perception. And finally, we used ScanNet, which handles 3D geometry quite well, as well as scene construction, and it de-emphasizes aspects such as semantic components. So these are the example response curves that I tried to fit to the data. The one that you previously saw, the one more indicative of a region that really favors novelty. A linear response, so modulation over varying degrees of similarity. And finally, a response that is most akin to what we would expect if a region can do pattern separation. And just to show you here, very clearly in the anterior hippocampus, we see largely that most of these regions show very clear favor towards novelty, and only the subiculum for the scenes shows a more modulated and some resistance to some feature overlap. And this very nicely actually aligns with our findings when we're using memory difficulty levels. So all of these perceptual, semantic, and geometric features kind of capture what we see in memory difficulty lower bins. When we're looking at the posterior hippocampus, we see that there are quite different patterns for the right posterior subiculum for objects and scenes. Specifically, we see exactly the expected pattern separation response profile for the scenes and the objects, depending on which type of method is being used. All of these model selections in gray, by the way, are based on the BIC, so Bayesian response criterion, to be more strict. But the blue ones I'm showing you are when we're using AIC to relax the model selection criteria somewhat. So in some cases, that means that the model would be selecting the more complex one. For the left posterior dentate gyrus, this is completely consistent across all methods, so a certain resistance to patterns overlap, feature overlap. And then for the right posterior dentate gyrus, we again only see these types of patterns when we're relaxing our model selection criteria. Again, this is very clearly all specific to scenes. So there is a tendency towards going with a more pattern separation-like response. So how does that match up with what we can see in memory? This actually is somewhat counter to what we see for the object. So here it seems to be driven more by the actual memory difficulty than for scenes, these other memory-independent features, so some differentiation. When we're looking at the cortex, most regions outside the MTL show this particular pattern, and most regions inside the MTL show a higher resistance to pattern similarity between lures and targets. But interestingly, the angular gyrus is really here one of the regions, or the main region in the wider cortical networks, that does show some support for a pattern separation-like response across these different networks, specifically for scenes, but not for objects. And again, this lines up quite well with the memory-based similarity bins, although there it's a bit less resistant to feature interference. But of course, everything that I've shown you right now is based on univariate analysis across all voxels of a given region. So what we're also interested in is applying multivoxel pattern analysis on a voxel by voxel basis to look at the response profile of a region. And so let's assume that this is the response profile for a given target. A region that is capable of pattern separation should be capable of altering its response pattern quite significantly, or potentially even fully, so that suddenly you see that these are rather dissimilar. But a region that is more biased, or that is not quite as capable of telling these apart, would show a much more similar response pattern. And this is identified here by the black circles. And our very preliminary data show that indeed the dentate gyrus does show this pattern, even in the multivoxel analysis, where it is capable of showing more dissimilar neural patterns of targets and lures, akin to what we would expect in pattern separation. So to conclude, target-lure similarity ratings based on neural networks are highly consistent in their association with brain responses, regardless of the models, of whether the models were trained on subjective ratings, semantic and visual information, or spatial geometry. The responses to neural network-based target lure similarity mostly align with the results obtained from memory difficulty bins, but specifically the posterior dentate gyrus shows some differentiation there by what it is most closely matched with for pattern separation response. Angular gyrus is more involved for pattern separation during scenes. And finally, we have some exciting preliminary results about the dentate gyrus and its ability to have separate patterns for targets and lures on a voxel by voxel level. So with that, thank you so much to everybody at the DZNE and to my funders and to everyone here.
Great talk. You can submit your questions either on the app or ask in person. Maybe while I'm waiting for the app, I can ask the first question. I was wondering about the performance of the classifiers that you use, the DreamNet and CLIP, just because the stimuli that you use in your task are not naturalistic photographs. They're computer generated, and so I just wanted to hear a little bit more about that.
Right. So you saying, I mean, we didn't necessarily look at how good they perform with regards to classifying new images, but it was more like looking at the feature embeddings and then correlating the level of similarity between target and lure pairs. So that is a bit different from what we could do with regards to how good the classifier is. But that's a good point. I will bring that up with the team. We should look into that.
Hi, thank you for the talk. Do you think the regions that's more has the pattern for pattern separation could be could be the downstream region that's receiving information from these regions that's basically aligned with the similarity gradient? So it sounds like it could be using those information and amplify the difference between...
That is an excellent question and one that we have asked ourselves quite a bit, and I would love to be able to actually find clear evidence whether it is an upstream or a downstream effect, but it's really, really difficult to do with fMRI. Just because obviously the directionality is this, it might be that angular gyrus is sort of the expression of the dentate gyrus pattern separation, or does it do part of the input? We can't actually tell. But maybe there is ways of doing effective or like dynamic causal modeling on an RSA-based approach. We'll see. We can try, but it's very difficult. We're out of time.
We're out of time. There is a question that maybe you said you have a poster, right? So
I do, yes, but that was different from, so all of this is different from the stuff on the poster.
Still find Helena at her poster to ask your question.
Exactly. Thank you so much.